Let me tell you a little bit about Professor Stephen Freeland. He's an evolutionary biologist. He took his first degree in Oxford, and then just to balance the books, he did a PhD in Cambridge. And then he went to a faculty position in University of Maryland in Baltimore County. But because he's married to a wife who works in the military, he had to have a hardship posting to Hawaii for a few years. But he's back in Maryland now, I gather. So it's a great pleasure to invite you to talk tonight. He's been doing research on origins of life, so very well suited to speak on the topic of will alien life share our genetic code. So over to you, Steve. Thank you.
And a very real thank you to you for coming. What I do is fairly obscure, and I'm happy with it, but I'm delighted that you've invested an hour of your time to come to me. The most important part really will be the questions from you. And just to encourage you, this is a broad-ranging topic, meaning that at any given moment, at best, some people are finding it basic knowledge, meaning that there are no dumb questions when we get to the end. It takes a certain amount of chutzpah to say I'm prepared to talk for an hour on the topic of whether alien life will share our genetic code. So judge me by that, because I think we can. I think within 50 minutes I can say something of scientific merit on that topic. But I put it to you that your questions to me will tell me whether I've done a good job. And before I go any further, because I really love this topic and I get carried away with it, let me just say a thank you to these four in particular. Poor Melissa, her photo didn't make it. But these are four students at various stages of young scientific careers who have invested enough trust to believe that this isn't a stupid question or a stupid way to spend their academic training. They're a wonderful crew, and I'll leave it at that. So it's a talk in 3 parts. I would be happiest giving a talk that was 50 minutes on part 3, which presumably is something to do with what brought you all here today. But I think that in order to get there safely, we need to get through parts 1 and 2. And without further ado, I want to begin here with a 10-minute biology primer. What is the genetic code? This is my PhD here at Cambridge. We're studying the genetic code, which we'll come to later. Some of you are biologists. Any of you with the most elementary college or even thorough high school education in biology will be at home with this topic. But I want to put it to you that this is worth 10 minutes, because if you're a biologist who knows this, I need you to understand how widely this is misunderstood and how important that is as we move forwards into the 21st century. To those of you who are not biologists, I want to stress to you how important this is, because if you understand this, you're now equipped to have a conversation with any biologist, any biomedical researcher that makes sense. And if you don't understand this, then you're not. It's that fundamental. So there we are. What is the genetic code? It is not DNA. And the popular press have gone so far down this line of talking about scientists have cracked the genetic code for this and that and the other, that it is becoming a real problem with our students and our lay audience who are convinced that the genetic code is DNA. It isn't, and until you understand that, everything is very confusing, and we cannot have intelligent debates about genetically modified crops or anything else that pertains to 21st century biology. We'll start here. A code, and I hate to use Wikipedia except when it's eloquent and correct, which it is here. A code is a system of rules for converting a piece of information, for example, a letter, word, phrase, or gesture, into another form or representation. One of the most famous examples that I feel we'd all be very comfortable with is Morse code. Morse code is not a string of dots and dashes. It is a rule book for telling you how to interpret those dots and dashes. The biological genetic code is the same thing. It is a rule book for telling you how to interpret DNA, or as we'll find out, RNA, into a fundamentally different chemical language that is constructed of amino acids. It is a code, it is an interface. It is not just a string of DNA. Now, I want to emphasize something very important here, which we don't really return to in the rest of this talk, but I'd be very happy to talk about, close to my own research, that the interest value of the genetic code for origins discussions is that it connects two utterly different, unrelated chemical languages, shown in cartoon form here, and biochemists in the audience can be duly horrified, but it's not that far off the mark, and we will see some slightly more accurate depictions before we're done. Two different chemical languages in the sense that at best there is dodgy evidence that these two like to hang out together. Just as there is nothing particular that connects dots and dashes to English symbols. And understanding how those two come into connection with one another is my best claim to turn up at an Origins of Life conference. I don't really study life's origins, probably. I do study how this came into being. This was one of the first decisions that life made on our planet. It has lived with us for billions of years, and it's an extraordinarily successful arrangement. And it's still a huge mystery as to how two unrelated chemical languages come into coexistence meaningfully. So, I want to walk through it in a little bit more detail before I'm done with the intro. I think that this slide has almost reached the end of its lifespan. 10 years ago this was necessary. Today I think that every audience is comfortable with the idea that genes are written out in a language of DNA, most famously depicted something like that as a rotating double helix, which is itself a sequence of letters drawn from an alphabet of size 4. Morse code, the dots and dashes, alphabet of size 2, dot or dash. DNA, alphabet of size 4, the famous G, C, A, and T that we see whizzing across the screen in Jurassic Park or countless forensic TV shows. Good. So there we are. Nobel Prize right there for understanding the structure and how that structure implicitly gives rise to replication, to evolvability, and to all that we associate with life. The other language is this language of amino acids. And amino acids frequently appear within health supplements and so on, but it's very important because it's very simple to understand what their relevance is. Here we have an alphabet of size 20 different types of amino acid in almost all organisms that are comprised of a constant backbone with a variable chemical side chain. Different colors here represent different types of atom. So there's a nitrogen and an oxygen and a carbon and a hydrogen and so forth. If you link these ones together, nose to tail, you form a different type of polymer, which, loosely, we can say is a protein. It becomes a protein because it will spontaneously fold up. And there's another Nobel Prize right there for understanding that there is enough chemical information within a sequence of amino acids for it to spontaneously fold up into a reliable, complicated, three-dimensional shape that has the right properties, for example, to catalyze a specific chemical reaction. Come back to that in a second. But just to link them together, without explaining how they came into coexistence, let's explain how they are in coexistence today. We need to find a way of linking two different chemical languages, and it occurs in two steps. The first step is known as transcription, where the double helix is unzipped long enough that a single strand of the DNA is read and copied letter by letter into a sister chemical language known as RNA. That process is transcription, because these two are very related chemical languages, and there is a one-to-one correspondence between the letters. Chemical details that separate them don't occupy us unless you want to ask about the RNA world towards the end of the talk. I mention that step because it's actually RNA which is then translated, meaningfully translated, not transcribed, into the other chemical language. In particular, a triple letter code word or codon is translated into a single amino acid, because you have a larger alphabet here. So just as in Morse code we need 3 dots in a row because we have an English alphabet of size 26, here we take 3 DNA letters in a row in order to specify exactly one of these 20 possible meanings. And in fact there's a class of molecule deep within molecular biology known as tRNAs, but you can just think of them as adapter molecules, whose sole purpose is to recognize at one end a code word written in the language of RNA and carry at the other end the corresponding amino acid. For example, in this case, the code word CAU corresponds to the amino acid histidine, and that's what this table has been telling you all along. So in blue we see all the possible 64 code words, and in green we see the 20 amino acid meanings, and a singular point of punctuation in the genetic language. 3 codons are assigned to the message, stop translating here, end of protein. That is the genetic code. That is fundamental knowledge to 21st century biology. Whether you're an ecologist, a conservationist, a biotechnologist, this is where it begins these days. If you've understood what I've said so far, you are indeed equipped to have a conversation with any kind of biologist. If you have failed to understand what I have said so far, or if you choose to believe that the genetic code is the same thing as DNA, then it can only lead to confusion. Thank you for indulging me for 10 minutes. Apologies to the biologists. But biologists, please take a moment to explain this because it's really important stuff. 4 Nobel Prizes in this. In fact, it's known as the central dogma of molecular biology because it defines everything. That's how we think of it. And I said I'd come back to these proteins, get enough of the right proteins together, and they will start to spontaneously interact in a complicated network that we know as metabolism. The set of reactions that sustain life lead to the creation of these letters, their polymerization into long strings, their interaction with one another, and cells and life as we know it. So proteins, the business end of life, DNA, the information end of life, and the code, the all-important interface between the two of them. Now, the reason that that's worth talking about, the reason that's so fundamental to any conversation with a biologist, and the reason why I think it's worth us talking about it for another 45 minutes today, is that superficially, life looks very diverse. Indeed, we can find non-biologists, such as Neil deGrasse Tyson, talking about the fact that aliens visiting Earth would be hard-pressed to find any commonality when we consider life's beautiful or inspiring diversity. But beneath this surface, life is all the same. In fact, even that diversity that I just showed you on the previous slide, which corresponds to a smattering of the organisms visible to the naked eye, represents just a tiny fraction of true biological diversity as we have uncovered it using the sequence data of DNA messages in order to build an objective tree of life. We find out that the fungi, the animals, and the plants represent one tiny twig in one of 3 fundamental branches. Using this technique, we discover that there is a fundamental branch known as the archaea or archaebacteria, which are as different from bacteria as bacteria are from fungi, animals, and plants. This is life's true diversity, and for the purposes of today, what's really exciting is that all of that diversity is united in a genetic code that originated some 3.5 billion years ago. All of it is using fundamentally the same genetic code. There are some very minor variations that have popped up in a few twigs, and they're instructive. We can get to them in the Q&A if you're interested. But we know that this code was in place at the point of connection between life's fundamental domains 3.5 billion years ago, a point we know as LUCA for the last universal common ancestor. It's one of the most fundamental decisions that evolutionary biology made in the history of life on our planet, was to set up this system. And it's been working with it ever since. Without going too far into it, we now know that particularly within these 2 branches are lineages that survive in a hitherto unimagined diversity of habitats. From pools of boiling water, to streams of acid, to lakes of alkali, to vacuums, to spent nuclear fuel, there are organisms that can thrive in those environments. And they're all using this same fundamental alphabet. It's a very good way to build life, apparently. So that's enough on what is the genetic code. Why should you care whether aliens will have a similar genetic code? I should be asking you that since you came here on a cold November night. But I'm going to offer an answer in the following format. This would be another hour lecture, and the typical one I give at public talks these days, thanks to the work in Hawaii. A simplified form of what's known as the Drake equation, which is one way to think about the number of times that life has emerged within our galaxy. We could take just 4 variables and multiply them together. The number of stars in our galaxy, the fraction of stars orbited by planets, the fraction of planets per star that can support life, and the fraction of these planets where life actually evolves. There are other ways to frame it. This is a useful one for now, because there is a paradigm shift underway within the natural sciences. From physics and astronomy through chemistry to biology, the clues are coming in that these variables lead to an answer far in excess of 1. Have we found life anywhere else? No. But the possibilities at this stage, and I emphasize this is a likelihood ratio rather than a binary truth, not truth, but a likelihood ratio is that there should be a lot of life out there unless we've missed something really big. It's possible that we've missed something really big. But we now know within the last 25 years that there are way more planets than was previously thought. This is probably the big news item. I'm sure many of you are aware of it. Almost by the week, the results, particularly from the Kepler mission, which is the first mission specifically designed to get an unbiased estimate sample of the number of stars with planets orbiting them, is leading many astronomers to propose that there may be an average of something like 1+ planets per star. Go back before 1995, and astronomers thought that that was closer to 0. Number 2, as I mentioned about these bacteria and archaebacteria that are not just surviving, but thriving in environments that we, until recently, believed were utterly inhospitable to life. Biologists are discovering, thanks to their new molecular toolkit, that life is way more diverse and tolerant of extreme conditions than we previously thought. We now know as an empirical fact that many types of microorganism can survive trips into space. In fact, it is entirely likely that NASA has unwittingly seeded other bodies in the solar system with microorganisms. One of the biggest dangers with life detection missions now is that maybe we took it there and then we discovered it. We'll see, we'll cross that bridge when we come there. But the reason that that is possible is not sloppy thinking so much as just we didn't understand this until recently. Number 3 is worthy of half an hour, it's going to get 1 minute. But life is made of the most cosmically abundant elements arranged in some of the most obvious ways. Life may seem very complex, and I'm not going to suggest to you that advanced biochemistry is not very complex, but advanced biochemistry is complex because it is about the detailed interaction of molecules. If you dig one level deeper and ask about the fundamental chemistry of biochemistry, most of life is made of just 6 elements: carbon, nitrogen, oxygen, phosphorus, and sulfur. I feel like I missed one in that. Anyway, schnapps. And with the possible exception of phosphorus, those are the 5 most abundant cosmic elements other than the noble gases. It didn't have to be that way. If we'd found out that life on this planet had come into being using some rare and unusual elements, that might be a clue that life is difficult. But it's made of the most abundant stuff that is out there, by orders of magnitude. When I say that it's the most abundant stuff arranged into the most obvious ways, I mean that some of the fundamental building blocks that I was introducing in part 1 are things that we know empirically now are spontaneously arising through non-biological processes as part of what we call astrochemistry. So I'm sure that many of you will have heard of Stanley Miller's spark tube experiments or studied them. 1950s, where he simulated a best guess at Earth's primordial prebiotic atmosphere, set up a very simple chemical apparatus, and within a matter of days was producing the famous organic soup. That was a good guess. It's been criticized on a number of grounds since, except for one interesting observation, which is that as NASA gets better at studying meteorites, which are themselves miniature laboratories of astrochemistry, it's finding results that are strangely, eerily similar to what Stanley Miller was discovering in the 1950s, including the fact that nucleobases and amino acids are reliable constituents of non-biological chemistry and are, to the best of our knowledge, quite literally raining down on the surface of every planet in the galaxy. That wasn't particularly well known until recently. And the last one is handing over to the geochemists and saying that with isotopic fractionation techniques, they are claiming, with increasingly widespread consent, earlier and earlier dates for signatures of life in the earliest rocks that we can find on planet Earth. Current estimates would place the origin for life somewhere between 3.5 and 3.9 billion years ago. And as far as I can tell, 3.9 is beginning to emerge as the winner. Which is interesting because it would appear that planet Earth was largely a molten ball of rock until something like 4 billion years ago. So in a remarkably narrow window of time, life arose. In fact, one of the primary phenomena that distinguishes those 2 time periods from a planetary perspective was, we think, the arrival of planet Earth's liquid water oceans, which is why the team in Hawaii that I worked with are very interested in understanding exactly when they arose and where they came from, because almost like a barren piece of yard that is watered appropriately, life mushroomed up. Put these 4 clues together, and it's starting to suggest to those who study such things that this equation leads to an n that is far, far, far larger than 1. And that's no longer based on the fact that there's a lot of stars. It's based on pretty much every clue that we can find. So, why should we care whether aliens will use the same genetic code? Because given life, what next? I prefer the term biocentric or life-friendly universe to any talk of an anthropic universe. Talk of an anthropic universe usually comes from cosmologists within the physical sciences who really mean a universe that is predisposed to give rise to life. Because to a physical scientist, a universe that is predisposed to give rise to life is a universe that is friendly to human existence. But there is a huge logical leap there between these 2 terms. We've just added one back into the Drake equation. Given life, what next? Given the fraction of these planets where life actually evolves, what would be the fraction of these where sentience evolves? Two unrelated variables. Very different answers. Really, what we're saying in this part of the talk is analogous to the question, if we were to arrive midway through this game and we could deduce something about the rules by which the pieces are moving, what could we then say with scientific rigor about the likelihood of this particular game turning out the way it did? That's the question that biologists, evolutionary biologists, can be asking to contribute to this view of the universe. And this is a good moment to pause with a slide that I'm sorry if you've ever seen me talk before. It's in every single one of my talks because I just love it. This is a slide that is from a relatively recent American textbook of biology, a fully secular, fully evolution-embracing textbook showing evolution as the textbook's illustrator chose to see it, spiraling upwards from 4 billion years ago through some of these bacteria and archaebacteria to increasingly complex organisms through the dinosaurs until it terminates here with humanity. And the question is, what's wrong with that picture? I suppose I'll keep it rhetorical today since you're all very dignified. What's wrong with that picture is that this is a white male. And this picture was drawn by a white male, and there is an innate tendency for human beings to perceive ourselves as being at the end of evolutionary progress. When you start to notice that that's what this picture has as subtext, you recognize that it really isn't a depiction of the evolutionary science that we've come to be studying over the last 150 years. It's a straightforward scientific veneer over a much older idea of Aristotle's scala naturae. An intuitive predisposition for humanity to think that there is an obvious order of progress and complexity in things as we encounter them. An idea that illustrated here in a medieval manuscript, which has added its own veneer of religiosity to find that God is the ultimate state of being, and actually, unlike Aristotle, who thought that non-being was the lowest state, we've added a whole inverse world of the diabolical. But that in between those, plants are obviously better than rocks, but less good than animals. Animals are less good than people, and people are just below angels and God. Rational philosophy from the golden age of Greece that has nothing to do with modern science, because Darwin's theory is recognized, at least in today's literature, as being the birthplace of modern evolutionary science, precisely by recognizing that people had been asking the wrong question about biological evolution up until that point. It's not that there was an absence of theories about evolution. For at least 100 years before Darwin, there were theories of evolution, but they were theories that thought the question was, what is the mysterious process that drives the scala naturae? What is the mysterious process that causes simple things to become more complex over time? Darwin's theory was the birthplace for an objective modern scientific view of evolution because it recognized that every species alive in today's world is the successful, equally successful product of 4 billion years of evolution. He wouldn't have known that number. But his theory is saying that there is no such thing as highly evolved. That is scala naturae thinking right there. And so it's deep irony that something like this icon is so deeply associated with the theory of evolution, far more in common with the theory of, say, Lamarck than with that of Darwin. Biological evolution, as we know it through science today, admits no mysterious purpose. In fact, I'm going to paraphrase Dawkins, who, as he often does when talking about evolution at least, is remarkably clear on this point, says, take an organism such as the beautiful seahorse, the leafy sea dragon. We tend to say colloquially that the leafy sea dragon is a fish that has evolved to look like seaweed in order to avoid being eaten by predators. All sorts of teleology in that statement. What we know we mean is that the ancestors of this fish that looked more like seaweed survived and reproduced more than their relatives who looked less like seaweed. No teleology, no mysterious driving purpose. So, when Darwin finally embraced that phrase, survival of the fittest, it was very clear that what he meant by fit was not some abstract notion of fitness as in health and well-being, musculature, some abstract notion of better, but rather a fit between the organism and its environment. Good and bad genetic variations exist only relative to a context, the environment in which the organism is competing for resources to reproduce. The fittest organism is simply the one that best fits, reflects, matches its environment. And you can sort of deconstruct an environment in your head with a GIS-type view into different layers, different components. Think about what each of those is contributing to the challenges faced by this organism to gather resources and reproduce, and start to deduce something about fitness, and now you're understanding what evolutionary biologists study when they study things. Now, this is, needless to say, a very, very complex topic, And then, of course, we can throw in all sorts of other things, from daily and seasonal variability to long-term geological processes such as plate tectonics that mean that today's tropical environment is tomorrow's Arctic environment, and vice versa. And start to understand that when organisms are evolving to fit their environments, not to ascend a scala naturae, this process is not exactly intuitively predictable as to where it will end up. And that's the point. This was a paper I wrote really to emphasize this point in response to advocates of intelligent design who relatively recently have come up with a claim that one big problem with evolutionary theory is that natural processes cannot create new semantic information. If that interests you, let's talk about it afterwards. Let's just say that it amounts to this. Biological evolution describes a natural process that transfers semantic information from a local environment into a chemical known as DNA. In that sense, we can say it's slightly analogous to the situation when gravity causes raindrops to form a puddle and the shape of the ground beneath that puddle becomes reflected in the underside of the water. If I could wave a magic wand and remove everything from this scene except for the water, I could deduce things about its environment. I might even apply algorithms that showed me there was significant semantic information, and I might confuse that for the signature of an intelligent designer. But I've missed the big picture, which is that this water, through a natural process, has inevitably come to mirror the information that was already present in the environment. And that's what we're looking at when we're looking at biological evolution. Evolution does not create information. It reflects images of the semantic information inherent to the physical universe. All of this beauty, all of this diversity, are partial reflections of the semantic information that is inherent to the physical universe. These images are beautiful, they're complex, they're unique, but they have not involved the creation of information in any way that science could measure or detect. Now, it becomes even more complex when we pause to remember that a significant fraction of any given organism's environment is other organisms that are themselves evolving. Broadly speaking, we can refer to this as the Red Queen theory of evolution, that says much of what adaptation is about is adapting in response to something else that is itself adapting. Pause for a second to reflect any of these pictures that did show up, and ask yourself whether you can notice what is the biological cause of the adaptation that you see. And now you're starting to think like a Red Queen evolutionary theorist, whether it's to attract mates, whether it's to disperse seeds, whether it's to catch prey, whether it is to warn off potential predators, or to fight off rivals in terms of mating. These images are bouncing off one another through geological time periods as they come down through the ages to the life forms that we find on our planet today. Predators, prey, mates, competitors, parasites, infections, all these are themselves changing and evolving entities that are contributing to the reflections of the physical universe that we see in life as we experience it. All of which leads us to the question, does evolution lead to any particular outcomes? And a 20th century orthodoxy that says, gosh, our chess game turns out to be very, very complicated. And I suspect the answer is that that doesn't lead anywhere predictable. So perhaps most famously, Gould would use the metaphor of replaying the tape of life back in the days that we remember when there were tapes. Would that lead evolution to the same outcomes? He says no, it would lead evolution down a pathway radically different from the road actually taken. Or we could expand it into more metaphysical statements that frankly go way beyond anything that science has a right to talk about in my mind. Man knows at last that he is alone in the universe's unfeeling immensity, out of which he has emerged only by chance. These are the sorts of views that come when we think that the game we've walked into is horribly complex. But there is a 21st century challenge at least being mounted in some quarters, including here in Cambridge by those such as Simon Conway Morris, who would be suggesting that far from its myriad of products being lucky and accidental, evolution is, in fact, remarkably predictable. The mistake we're making up here is the classic mistake, the mistake of Aristotle, which is mistaking common sense for scientific fact. It's very complex, and it may be way more complex than we ourselves can understand in terms of predictability and outcome. That's not the same thing of saying it lacks predictability in outcome. So, what leads these folks, such as Vermeij and Conway Morris, to make the statement that evolution is indeed predictable? Empirical evidence. This, again, a beautiful illustration from Dawkins' Blind Watchmaker, but I want to show you this time 4 independent lineages of animal that have each been confronted with the same environmental stimulus to swim efficiently through deep water and have all evolved the same fundamental morphology, the bullet shape, because that's an efficient, low-drag model for swimming through water. When we see independent arrivings at the same morphology, from different lineages that have been faced with similar challenges, then we can infer some measure of predictability. This is the sort of shape that you will get from a large multicellular organism challenged to swim efficiently through deep water. Broadly speaking, that represents a whole class of evolutionary study known as comparative studies, where you're looking precisely for this sort of signature of predictable outcome in order to understand the adaptation that you're seeing. And it extends beyond such things into places where we wouldn't have expected it. For example, two different fundamental groups of mammals, the placentals and the marsupials, and it's long been recognized, but largely written off as a curiosity, that there are a number of eerie parallels from lineages whose last common ancestor was probably something like a nocturnal tree shrew. The most interesting one of these, just for face value, is we've all heard of saber-toothed cats. What's not widely understood is there were actually not even two, but three independent arrivals from evolution of a saber-toothed cat, including two placentals and one marsupial, if I've got that the right way round. Twice over, evolution, 3 times over in that case, has arrived at something that we would not have been able to predict in advance was in any way a logical outcome of fitting an environment. And then we start to realize it applies to things as bizarre as anteaters or flying squirrels. And we start to recognize that our inability to predict where evolution is going should not be mistaken for evidence that evolution is going nowhere. Now, Simon Conway Morris would be an advocate, and believe me, he can go way beyond this, and I'm sure he's spoken here before, and I encourage you to go and see him if he speaks again. He can go way beyond this with examples that stack up into a story, suggesting that, in his mind, it's actually the pendulum has swung the other way. He firmly believes that evolution is, in fact, predictable. And something analogous to a scala naturae might actually be a better fit for the natural sciences as we encounter them. I was asking earlier if he's here. He knows I say this, but I say it to his face. I wouldn't go that far. But I would suggest that it's an open debate. And that at the very least, when we look back at this 20th century orthodoxy, we should... Oh, absolutely. Sorry, I get excited and I speak too fast. Let me calm down. I would suggest that this orthodoxy goes beyond the evidence, the empirical evidence at this stage. That's all I want to assert. All of which brings us neatly back to the question of the genetic code. This idea of finding convergence and using it to infer predictability within biological evolution is a good idea, but it relies on having independent lineages that have arrived at more or less the same point. If we really want to understand something about the fundamental nature of life, then our problem becomes that all of life has the same fundamental basis. Genetic coding, in as far as we're able to detect today, has arisen once. And one genetic code, with some very minor variations up here, one genetic code is present in every organism. So there's no possibility about asking questions relating to convergence when it comes to genetic coding. So I would simply like to compliment Simon Conway Morris's approach to what I consider to be a very important question for 21st century evolutionary biology by saying that in addition to comparative approaches, which are looking for convergence, there is a whole other school of evolutionary theorizing from the 20th century that tends to get overlooked. Really, I'll say it's as a result of bioinformatics, and if you care, ask me. If you don't, just accept this one is the underdog these days. But optimality studies ask a different question to infer predictability. They ask whether observed biological features match some a priori concept of an optimum. That might be human design, and that's what I'm attempting to illustrate with this slide of an early submarine design, where thanks to a wind tunnel and physics, engineers were able to predict the same sort of bullet shape. The important point is we have an a priori concept of what would be best or most efficient, and then we ask whether nature matches that optimum. That approach works very well when we want to ask questions about life's fundamental characteristics. So, finally, part 3. What can science legitimately say about the inevitability of life's genetic code? Which is a different way of asking, will aliens have something like our genetic code? Why this particular genetic code? This is where I began at Cambridge as a young PhD student. My supervisor put in front of me, actually, sorry, that's not a slide. There we go. My supervisor put in front of me an old observation from 1965 from one of the pioneers of molecular biology, that amino acids are not assigned to their genetic code words at random. They're assigned in a pattern such that similar code words encode amino acids with similar properties, particularly the property of hydrophobicity. Here's one particular measure of hydrophobicity known as polar requirement. The 20 amino acids are aligned quantitatively, and for the sake of visual identification, the hydrophobic amino acids are shown down here in blue, hydrophilic amino acids are shown up here in red, and the in-betweenies are shown in green. Here they are again mapped onto the genetic code, and intuitively that's not a random genetic code. So my supervisor encouraged me to write a computer program that could build on an earlier observation he'd made. Oh, sorry, just before we get there, one other important fact. Hydrophobicity is known to be a primary factor in causing these proteins, these long polymers of amino acids, to fold up reliably into a particular shape. So we know that it's intimately associated with the adaptive value of an amino acid sequence. And here we're seeing that it's distributed non-randomly in the way it's encoded. So he asked if we could write a simple computer program that would calculate the average change in hydrophobicity caused by every possible mistake, mutation or misreading or misdecoding that could occur within a given genetic code. For example, the code on UUU, which should encode phenylalanine, could be miscoded as UUG and give rise to leucine. That would be a mistake. With a measure of hydrophobicity, we can quantify that. And we can apply it to all of the codons, all of the positions within the code word, and come up with something I'm going to call an error value for the genetic code, which is a magic number representing something of its susceptibility to genetic errors. A high error value would indicate a genetic code is very vulnerable to genetic errors. An average genetic error causes a big change. Low error value means it's very robust to genetic errors. We can then use the same computer program to take a genetic code, strip of its meanings, and reapply them at random to create a large sample of random genetic codes and measure their error values, alternate realities which biological evolution, as far as we can tell, has not chosen as outcomes. And if we do that, and I should emphasize that we've been very conservative here, all we've done is recognize all the molecular machinery as we find it in life, but suggest there's no logical or biochemical reason why any one particular meaning has to be at the end of any one particular adapter molecule. Create a large sample of alternative codes that we think are completely physically and chemically plausible but not biological, measure their error values, and plot out the results. We found this result, published back in 1998, where we see the error values of all of our theoretical codes, 1 million of them, that we generated down here. We find the frequency with which they occur in our random sample, and we get something like a nice bell curve. And we find that the actual genetic code that nature settled upon 3.5 billion years ago has a remarkably error-robust architecture. In fact, in our sample of 1 million random genetic codes, we found one that outperformed nature's choice in terms of its resistance to average decoding errors. That was a paper that generated a lot of success. I should emphasize from the get-go, firstly, that all we'd done was quantify and test an observation that came from a giant of molecular biology decades before. Secondly, we weren't the first, and we certainly aren't the last with this observation. That one is just a review article dating back to 2003 in which we cite dozens of papers that had tried to tackle the same question. So I'm telling you about that version of it because that's the one you'll often see. For whatever reason, the internet loved this one. You'll often see it. I don't think for a minute we actually created that idea. I think at best we contributed to measuring it. And the story continues. You can just go, here is one of the recent examples that's still trying to understand what that result means. We would still maintain that that is a signature of predictable evolution very early in life. That the way the genetic code is organized was, if not inevitable, then at least somewhat predictable in terms of fundamental physics and chemistry and processes of mutation that we can understand a priori. Now, this is where I tend to give Simon Conway Morris a hard time, and it's where I first connected with him. In his first book on the topic of predictable evolution, where he was saying that far from its myriad of products being lucky and accidental, evolution is in fact remarkably predictable, Simon Conway Morris early on cites this work to say aliens would have the same genetic code. And as the author of said work, I would say, no, it doesn't say that at all. At best, it's the beginning of an insight that is starting to open up the debate, moving away from a 20th century orthodoxy that it's obvious evolution is unpredictable. So, what have we done since? Very, very restricted test that, as I say, simply permuted the amino acids at the end of these adapter molecules and asked if other architectures that held all of the other variables constant would give us worse or better genetic codes. A next logical question is to go one level deeper and ask why this particular amino acid alphabet. Biologists grow up thinking that there are 20 types of amino acid. Chemists know that is not true. Chemists know that an amino acid looks like this in its secondary structure, a fixed backbone and an utterly variable side chain. We know of 20 biologically encoded amino acids, but technically there are an infinite number of amino acids that are possible. In fact, we can do more than that. Oh, and I see here, we're just widening out our chess question. Now we're arriving at a game, but trying to change the very rules by which it's played, perhaps the very pieces that are involved, and asking whether we can detect that the game that we are encountering carries deeper signatures of predictability. I can say that this isn't just a theoretical construct, but something that I think is worthy of more attention than GMOs, in my humble opinion, is the fact that biotechnology is now learning to insert extra amino acids into genetic codes of living organisms in order to build proteins that have never before seen the light of day. They're getting so good at it that even back in 2010, this review article was talking about a collection of slightly over 70 unnatural amino acids that have been incorporated into genetic coding. And if you scratch the surface on this, I think it is more than coincidence that in the 1990s, in the dawn of the genomic sequencing era, enterprising biotechnologists were finding out in the Supreme Court that they could not patent proteins that they had discovered, which evolution had created. It is interesting, to say the least, that some of those same individuals are the ones who've gone off and learned the technology to insert amino acids to create proteins that nature has never created, which, according to that Supreme Court ruling, would be eminently patentable. What I think is worthy of debate and discussion is the fact that at present we have almost no guiding theory for what is going to happen when we start doing this large scale. But I think it's important. Why? Let's go back to Stanley Miller. I've already mentioned to you his famous experiments where he produced about half of the amino acids, half of the 20 amino acids that are still at work in your genetic code today, when he simulated what he thought was prebiotic conditions. I've already told you that meteorites match this eerily well. And in fact, this table is a small version of a much larger table I could show you, showing enormous agreement that about half of the amino acids still at work in your body today are almost definitely produced abiotically and delivered to every planet in the cosmos. That much at least is beginning to get penetration within a broader audience. But there's something very important here. Well, I'll come back to that. Oh, sorry, that's just telling you that there's an awful lot of this stuff coming down. There's a classic example of the meteorite showing the amino acids that have been found. What I want to draw your attention to, though, is something different, which is that an awful lot of amino acids that are not part of biology are also found in meteorites, are also produced by Miller-like spark tube experiments. So not only are the synthetic biologists showing that it is technologically possible to enlarge and change the amino acid alphabet, the universe is showing us that lots of other possibilities were ripe for incorporation into an origin of life. Now, that only accounts for half the amino acids in your genetic code, and to cut a long story short, it's becoming increasingly clear that biology itself invented the remaining pieces, the remaining 10 amino acids that make up the 20 in your bodies. In fact, we see something very much like that going on with a 21st and a 22nd amino acid that are currently entering the genetic codes of some of those bacterial lineages. Some of the more interesting changes that we've noticed recently are going on in genetic coding. That's important because a long-standing idea tells us that the abiotically plausible amino acids, those that crop up in meteorites or spark tube experiments, typically are found at the head of the biosynthetic pathways by which life is currently synthesizing the other amino acids. This idea suggests that we find the yellow abiotic amino acids here precisely because this modern metabolic pathway that takes an abiotic amino acid and tweaks it slowly into a biological amino acid, this is a footprint of very, very deep evolutionary history, showing us the very process by which early life invented the remaining amino acids. And I draw your attention to that just to add to this concept of a much larger pool of possibilities than those that life ended up with. These are the amino acids that we think life invented. And even if we take at face value this sort of pathway, then we find located throughout it other legitimate amino acids such as homoserine and cystathionine, which are not used in biological coding, but would clearly have to be invented in order to get here if we're right about this view of the world. In fact, this was a list that we drew up a couple of years ago just showing some of the other amino acids that must have been available to genetic coding but were stepped over by evolution in order to arise at the code as we know it. All of which says that we can get something like a good set of possibilities extending far beyond the 20 genetically encoded amino acids if we want to ask questions again about whether these 20 were in some way predictable. Here I'm just going to use one experiment that we've done with the Murchison meteorite, which has been analysed very carefully, and the biosynthetic list that I just showed you. We identified that although it's hard to come up with a priori reasoning for what makes an amino acid set valuable, it isn't hard when you talk to biochemists to recognise that we think that hydrophobicity, charge and size are three very important properties of these molecules in terms of defining what they can do, what proteins they can build. We just launched a simple adaptive hypothesis that said what would make a set of amino acids better or worse for building proteins, and suggested the following two criteria for a set. Number one, in any given property, such as hydrophobicity, we might say that a better set covers a larger range. But counterbalancing that, we might also suggest that a better set of amino acids should be evenly dispersed across that range for size or charge or hydrophobicity. It would take a long time to really go through that in detail, but the analogy here is to challenge you what would be a good set of building blocks to use if all you know is that for the next 4 billion years I'm going to challenge you to make stuff, and whoever makes the best stuff wins. This allows you to approximate almost anything that you would want, mathematically better than any other solution I could think of. But it's just a hypothesis. There's our adaptive hypothesis. We propose that a good or well-chosen set of amino acids would have a larger range and more evenness in size, charge and hydrophobicity than would a random selection. And then we return to our favourite. We just go back to the computers. We take a pool of amino acids defined according to what we find in meteorites or what is on those biosynthetic pathways, however we choose to ask the question. We select random samples of them, measure their range and evenness in terms of the properties we think are important, repeat for a large number of replicates, form our bell-shaped curve, and ask where life lies on this selection of possibilities. And cutting to the chase, rather than show you the detailed results, no matter how we ask this question, when we measure the properties of a set of amino acids chosen by evolution, the ones that are in your bodies now, we find that random chance would be highly unlikely. Between 0 and 0.1% is our best estimate according to how we ask the question. It would be highly unlikely that randomness could represent the chemistry space, the range and the evenness, as well as biology does. Again, I stress that we don't think that we've found that nature has chosen a determinate set of building blocks. We do think that we've stumbled across a strangely strong and simple clue as to a predictable feature in life's most fundamental biochemistry. We say in our conclusion to that paper, 2011, this is at best a start point to thinking about the presence or absence of amino acids within the standard genetic code. This is where I think we're at. I think that was what we picked up with a remarkably naive and simple protocol. And currently, we're trying to distinguish, just basic scientific process, whether actually we can really pick up a signal that we believe in, or whether we can explain it away as an artifact. And in 1 minute, I'll just show you, these are the papers that are coming out this year that are pushing us forwards on this. We think that using our same set of logic and predictions, we can apply it to the 20 amino acids found in your genetic code, and to the concept of 10 that were early and 10 that were late, and find exactly the logic that we would expect if everything I've told you is true. That's a paper in Journal of Systems of Chemistry. We think we can use computers now to generate an exhaustive, a mathematically exhaustive set of legitimate amino acid structures to enlarge our context for asking this question, so that we're not reliant on samples from meteorites. That one is one that's coming out right now in the Journal of Chemical Informatics. We think that we can get a lot more sophisticated about the protein building potential of non-biological amino acids. And that's a paper that we've just published in JME. We're still a step away from saying what can you do with amino acids. In fact, that's a very difficult question. But we think that this gives us a foundation for asking questions that not only interest me in terms of this talk, but should interest you in terms of biotechnologists who are about to unleash a new generation of engineered proteins. And we think that we have at least some potential insight into a deeper question that I haven't even gone to today, which is why would life invent two different chemical languages? Perhaps the most overlooked question of early evolutionary biology. It's not at all clear why evolution would arise at two independent languages and a complicated system for interfacing them. I can do a handy-wavy thing that talks about an RNA world and the fact proteins are better catalysts, but all the clues are coming in that RNA can do a surprisingly good job at catalysis. Basically, that argument doesn't add up, but I'll leave that for questions. This paper said something different, and it comes from mathematics and computer science that suggests that the ease with which you can solve a given problem may depend heavily on the language in which you express that problem. And that by creating two separate languages and an interface to join them would be one very powerful way of tweaking the language relationship into a form that would best allow you to solve the challenge of building modular molecules over 4 billion year periods. Very, very baby step on that front, but for me one of the most interesting ones, and I'd love to find more people to work with on that. But having said that, let me finish there, except to say thank you for your attention in a warm room and a soft voice. I have deliberately not said anything yet about my own personal take on whether theology lies in all of this. Over time of talking to institutes such as the Faraday Institute, I've learned that my personal theology is much less of interest to you than the science that I can talk about. But I'm very happy to answer questions about why I, as a committed Christian, think that this is valuable work and where I happen to find my theological significance in all of this. So thank you very much, Q&A.